Okay, hello. I'm Ryan Williams. I'm going to talk about work I was a part of at Mount Sinai in New York for parallel analysis of single-cell RNA-seq data. When I talk about single-cell data, I'm basically starting from the gene expression matrix, which is like a giant sparse matrix of cells by genes with just a number indicating how much each cell in a population was expressing a given gene. We have increasingly large data sets of this form. There's a whole, upstream processing part involving going from reads to these matrices that I'm just assuming is standard. I'm not covering that here, but so there's, the largest public data set is like 2 million cells by 30,000 genes. So these, they're starting to be pretty large. And if you want to do some of the standard processing on these things, it gets unwieldy on large cloud instances. They still kind of take a long time. There's a lot of opportunity to speed up the analysis and scale it out to larger data sets. Most of the things people are doing with these are different kinds of filtering and normalization or clustering to identify cell types. There are a wealth of tools in Python and R that are popular and people use that do this effectively, but as I mentioned, they can kind of struggle with the increasingly large data set sizes. The data is often persisted in various text-based formats or different schemas embedded in HDF5, which I will talk more about. One of the first things that we did in order to scale these analyses better was to use an alternative to HDF5 called czar. That is like a new project from in the Python ecosystem, but it has implementations in several languages at this point. Both HDF5 and czar essentially give you ways to store multi-dimensional arrays and even groups of them in specific like schema kind of hierarchies. HDF5 stores them in single large files on some underlying file system. And that poses some problems for parallel writing them, for sure. And it's also a bit difficult to read from these files in parallel. And when I say in parallel, I mean either just using many cores on one machine or like hundreds or thousands of machines in a cluster all pointing at the same data, but that's also been chunked and spread across machines to support that kind of access pattern. HDF5 is also hard to use directly in like public clouds, just as an artifact of how it's implemented. So czar is a lot simpler and solves some of those problems. It basically materializes the chunks of these multi-dimensional arrays as individual files on some underlying file system like abstraction, which can be your local disk or a cloud store. Or even like a database that's just storing binary blobs and exposing them in this kind of structured format. It comes with some simple metadata. It tells you where tells you about the shapes of the arrays that the chunks are representing, and the chunks themselves are just like, gzip or other compression codecs applied to the literal subset of the higher dimensional array. So we added support for that to ScanPy, which is a popular single-cell tool, and we're able to use that downstream with various parallel processing frameworks. BOSC in particular is a easy way to get multi-core or cross-machine parallelism in Python. We also made a library called zappy that basically does the same thing for different other parallelization frameworks like Spark, Beam, and Pyren, which are interesting. You can look up. They basically just give you ways to take code that's written against a NumPy array interface and parallelize the operations that you're doing to those arrays. So we added it to the data storage layer of ScanPy, which is called ANData, which is like a way to have annotated data matrices with metadata about rows and columns. Here you have some simple code example of basically reading some single cell data that's stored in the czar format and then doing like parallel processing on it using BOSC. some obligatory benchmarks. You can find, I think my poster's still up where you can talk to me afterwards, but the point is, like, as we expect in these kinds of things, like, you often pay a cost for parallelizing, like maybe the total amount of CPU you use is like 4 times higher, but you're able to use hundreds of cores potentially, so you can speed up your runtimes by arbitrary amounts, kind of. And we're working on pushing that parallelization further downstream in the analysis pipeline to more complicated algorithms like clustering. Today it works for simple pre-processing phases. So that's all. Thanks.
Okay. Thank you very much, everyone.